All right, let's dive in. Today, we're going deep into the world of chromatography.
Chromatography, yeah.
Mm.
It's really exciting stuff, especially with all the recent innovations happening. HPLC, UPLC, SFC. These techniques are really shaking things up in labs.
They really are. I mean, think about it. They're used everywhere, right? From figuring out what's in a complex mixture to finding those tiny, almost invisible impurities.
Exactly. And they're making lab work way more efficient, which is huge,
Yeah.
especially in areas like, drug discovery and development. That's where these advancements become super crucial, right?
Absolutely. When it comes to something like new medicines, getting it right and getting it right quickly is so important.
Couldn't agree more. Speed and accuracy, that's the name of the game.
Okay, so for this deep dive, let's really get into the nuts and bolts of it all. We want to understand not just what these improvements are, but also the science behind them.
That's the plan. We're going to pull from the research papers and really dissect how these techniques work and why they're so groundbreaking.
But we want to keep it engaging, right? Like a good conversation, not a dry lecture.
Absolutely. So to kick things off, let's just do a quick recap, a little refresher on the fundamentals. What exactly is chromatography all about?
Yeah, good point. Let's set the stage.
At its core, you can think of chromatography as a powerful tool for separating the different parts of a mixture. Like, imagine you have a bowl of mixed nuts. Chromatography would be like sorting those nuts by type so you can look at each one individually.
Makes sense. It's like organizing chaos.
Exactly. Now, remember those three main techniques we're focusing on: HPLC, UPLC, and SFC.
Right. High performance liquid chromatography, ultra performance liquid chromatography, and supercritical fluid chromatography.
Those are the ones. And what's really cool is how they've been evolving to be not only faster, but also way better at telling those subtle differences between substances. We call that resolution.
So let's talk about UPLC. From what I've read, it's like a speed demon in the world of chromatography. What makes it so fast?
Well, the secret sauce is in those columns it uses. They're packed with super tiny particles, much smaller than traditional HPLC. Now, to push liquid through such small spaces, you need some serious pressure.
I can imagine, like trying to force honey through a coffee stirrer. So what's the benefit of all that pressure?
It's a bit like this, right? That higher pressure, combined with those teeny particles, makes the analysis way faster. But even more importantly, it dramatically improves the resolution. It's like switching from an old blurry photo to a high-definition image. Suddenly you see so much more detail.
Ah, so it's like having a super-powered magnifying glass for separating and seeing those individual components, and you get that level of detail much faster.
Exactly. And remember what we were saying about wanting to get knowledge quickly but accurately. That's where UPLC shines. More information in less time. Now, another really important aspect, especially in those sources about impurities, is sensitivity.
Right, sensitivity. Being able to detect even the smallest amounts of a substance. That's critical, especially when you think about things like impurities and medications. You don't want any unwanted surprises lurking in there.
Absolutely. What's really neat here is that it's a team effort. We have innovations in both the detectors and the columns themselves, leading to a huge boost in sensitivity.
So it's like having a super sensitive radar able to pick up even the faintest signals from those separated components.
Exactly. The detectors are now much better at capturing and amplifying the signals, and the columns are designed to interact more strongly with the substances we're interested in.
And this isn't just some theoretical advantage. It has real-world implications for safety and quality control, particularly in pharmaceuticals.
Absolutely. Being able to identify and measure even the tiniest amounts of impurities is vital for ensuring the safety and effectiveness of medicines. You want to be absolutely sure that the active ingredient is at the right level and that no harmful contaminants are sneaking in.
Now, you keep mentioning columns. What are some of the cool advancements in column technology that are driving these improvements?
The materials inside the columns, what we call the stationary phases, have come a long way. And the materials used to make the columns themselves, they've also gotten a major upgrade. It's not just your basic silica anymore. We have a whole array of specialized materials, each with its own unique properties.
So what kind of differences are we talking about here? Like what makes one column better for separating certain substances than another?
Think of it like this. We now have columns that are specifically designed to interact with certain types of molecules. Some are great for separating based on size, others on charge, some on whether a molecule likes water or oil. And there are even columns that can separate those mirror image molecules, the chiral ones.
Mirror image molecules. I'm intrigued.
Yeah, they're called enantiomers, and they can have completely different effects in the body. Imagine a drug where one enantiomer is beneficial, but its mirror image is harmful. It's crucial to separate those out.
That's fascinating. So it's like having a whole toolbox of specialized filters, each designed to catch a different type of molecule.
Exactly. This gives scientists so much more flexibility and power in tackling those complex mixtures. Now let's switch gears and talk about SFC, supercritical fluid chromatography. This one always felt a bit different to me.
It does seem like the odd one out. Why is that?
Well, unlike HPLC and UPLC, which use liquids as their mobile phase, SFC uses something called supercritical fluids.
Supercritical fluids, that rings a bell. Isn't that when a substance is kind of like both a liquid and a gas at the same time?
You got it. It's when you heat and pressurize a substance beyond a certain point, and it takes on this unique in-between state. And that gives SFC some pretty neat advantages.
Like what?
Well, for certain types of molecules, especially those that don't like water, SFC can be even faster than liquid chromatography. It's often considered a greener technique because it uses less of those harsh organic solvents that can be harmful to the environment.
So it's faster and it's better for the planet. Two big wins. And I remember reading that SFC is particularly good at separating those chiral molecules we were just talking about. Why is that?
Chiral separations are definitely one of SFC's superpowers. When you combine SFC with those special chiral columns we mentioned, it becomes incredibly good at separating those mirror image molecules, which is essential in drug development to ensure you have only the safe and effective form of the drug.
Okay, we've covered a lot of ground in terms of the how. We've talked about the tiny particles, the high pressure, the fancy columns, and the supercritical fluids. Now, let's really dig into the why. How are these advancements actually being used in labs? Can you give us some real-world examples?
Absolutely. This is where the rubber meets the road. Let's go back to drug discovery. Chromatography is used at practically every step of the process.
For example, when scientists are creating new drug candidates, how does chromatography fit into that initial stage?
Well, right after a new compound is synthesized, you need to make sure it's actually what you intended to make and that it's pure. UPLC, with its speed and resolution, is perfect for quickly checking the purity and identifying any unwanted byproducts from the chemical reaction.
So it's like a quality control check to make sure there are no unexpected ingredients in your recipe.
Precisely. And as a drug candidate moves through the development pipeline, you need to figure out how it behaves in the body. How is it absorbed, distributed, broken down, and eliminated? All those ADME properties.
Right, ADME. Those are crucial for understanding how a drug works. How do these advanced chromatography techniques help with those studies?
Well, we often combine chromatography with something called mass spectrometry. It's like a dynamic duo, LC-MS. This combo allows us to measure drug concentrations in things like blood, urine, or tissue samples with incredible accuracy. That way, we can see how the drug moves through the body over time and understand its safety and effectiveness.
So it's like tracking the drug's journey through the body? That's amazing. What about those metabolites, the substances the drug is broken down into?
That's another key area. Chromatography helps us identify and measure those metabolites. Understanding a drug's metabolism is super important because sometimes those breakdown products can be more active, less active, or even toxic.
So it's like following the drug's transformation as it interacts with the body. And the fact that these new chromatography methods are faster and more sensitive must make a huge difference.
Absolutely. Faster analysis means quicker results, which
Yeah.
mean faster decision-making in drug development.
Hmm.
And being able to detect those tiny amounts of metabolites gives us a much more complete picture of what's happening.
Earlier, you mentioned that these techniques are used in other fields too. What are some other areas where these chromatography advancements are making a big splash?
You're right, it's not just pharmaceuticals. These techniques are also crucial in environmental monitoring, food safety, and even clinical diagnostics. Basically, anywhere you need to separate and analyze complex mixtures accurately and efficiently, you'll find chromatography playing a key role.
Okay, so these advancements are clearly super powerful. But are there any limitations to these advanced chromatography techniques?
That's a great question. It's always important to consider both sides of the coin. While these techniques offer huge advantages, there are some challenges too. For one, the cost of these high-tech systems can be a barrier for some labs.
Makes sense. Cutting edge usually means a higher price tag. What about the methods themselves? Are they more complex to set up?
Yeah, method development can be quite tricky. Figuring out the best combination of solvents, columns, temperature, flow rate, sports. It can be a bit like solving a puzzle, especially with really complex mixtures. It takes expertise and time to optimize those conditions.
So it's not just plug and play.
Not quite. There's still a lot of know-how involved in getting the best performance out of these systems. And some types of samples can be a bit finicky. For example, really thick or sticky samples might not play well with UPLC because of the high pressures involved.
Okay, we've covered so much ground today. Let's do a quick recap of the key innovations in chromatography that we've discussed.
We talked about UPLC and how its smaller particles and high pressures lead to incredible speed and resolution. We explored the impressive gains in sensitivity thanks to better detectors and columns. And we discussed how the development of those specialized columns allows scientists to separate a much wider range of molecules. And of course, we delved into SFC, the technique that uses supercritical fluids and really shines in chiral separations.
And what are the big takeaways for labs, especially in the world of drug discovery?
Well, these advancements make everything faster and more efficient, saving time and resources. The improved resolution gives us a much clearer view of those complex mixtures, and the increased sensitivity allows us to detect even the tiniest amounts of impurities and metabolites. It's all about getting more detailed and reliable data, which is crucial for developing safer and more effective medicines.
This has been a truly fascinating deep dive into the world of chromatography. It's amazing to see how these techniques are constantly evolving and driving progress in so many important fields.
I agree. And as these analytical techniques continue to advance, it raises a really interesting question. How will our ability to understand and manipulate those complex chemical and biological systems change in the future? Think about it. Even faster drug discovery, more precise diagnostics, a deeper understanding of the molecular world around us and within us. The possibilities are pretty exciting.
They really are. And with that thought-provoking question, we'll wrap up today's deep dive. Until next time.